Check 1, 2, 3. 1, 2, 3. 1, 2, 1, 2. Check 1. Okay. Check 1, 2.
While we're waiting for a few additional people to grade the elements, I'll get us started. I'm Dr. Paul Heisner from the Department of Anatomy and Cell Biology. Across the river, very appropriate thing for the day. We welcome you to the second in our Geneva lecture series of this current academic year with pleasure on behalf of the Geneva Lectureship Committee, on which I and others do serve. On behalf also of the Program in Literature, Science, and Arts, which is a co-sponsor of this lecture, and the University Lecture Committee. We're most indebted to all of these groups who have joined us to bring an outstanding scholar to campus to provoke our thoughts and our response to very stimulating books and discussion that she will be bringing to us. Many of you, I notice, are familiar with the Geneva Lecture Series from past experiences. I would bring the attention of the series to those of you who may not have joined us on previous occasions as one which has brought a broad series of thinkers, intellectuals, Christian philosophers to the campus to stimulate discussion and our thinking about the integration of faith and learning. We're particularly eager to get on with our proceedings this afternoon, but we hope that you will take out maybe, as mine became, even walking down the aisle, a rather soggy questionnaire toward the end, and we'll probably remind you once again about this, is that we would appreciate very much your suggestions as to future speakers, and we would appreciate your responding and maybe giving one of these to one of the ushers on the way out. Before turning the podium over to Dean Sims, I would like to remind you of the opportunity to interact with our guests again this evening. We'll be able to enjoy a lecture, Science Stumbles on Design, at 7:30 in the Buchanan Auditorium in the Papa John Business Administration Building. So we encourage all of you to attend that, and we will explain perhaps there, as well as a brief mention here of an opportunity to meet with Dr. Behe during the Saturday morning hours before the game in the Union, and we'll say more about that. Well, without further introduction, except from Dean Leslie Sims, Dean of the Graduate School and Professor of Chemistry, which is Dr. Behe's discipline as well, I'd like to call him forward as a working administrator. A lot of us on the faculty have great admiration for Dean Sims, and he insists on taking a hand in teaching, I believe, within the spring semester usually, whenever he can.
I don't have to explain it.
Well, maybe I've let him into things here, and I'd like to call Dean Sims forward to tell us how we will proceed and introduce our guests and our panelists.
I guess I'm called on to explain that in the first 10 years or so as an administrator, starting in 1978, I insisted on keeping a research program going, moved it from one university to another, and after 5 years of reestablishing a laboratory and having students, I reluctantly decided that it was unfair to students to continue to do research, but I continued every semester until last year, when I was asked to take on new duties in the provost office, and so I did not teach last year, but this year I do hope to get back to teaching because it's one of the great joys of being series. It left off the first line, which is intelligent design in biochemistry, criticisms and responses. You now know what you're getting into. Michael Behe is, above all, a brave man. He has in his latest book, which I have a copy of here if anyone would like to see it, Darwin's Black Box, confronted the acentral dogma of biology, that Darwinian evolution is sufficient to explain the origin of life on Earth, the remarkable diversity in which life is presented through species, and the adaptation and natural selection of life forms in response to environmental factors. There are understandably differences in the reaction of various persons and groups to this book, but much greater unanimity that the book is, in the paraphrased words of reviewers, a lively exposition which conveys a sense of biochemistry's hidden beauty, and, perhaps less agreed to, a thoughtful statement of the biochemical challenge to Darwinian evolution. Michael Behe is professor of biochemistry at Lehigh University. He received his PhD degree in biochemistry from the University of Pennsylvania. He will, in his presentation, have about 30 minutes, maybe a little bit more, to present an opening statement about his work. We then have responses from two guest speakers who will take about 10 minutes each in response. Professor Behe will then have an opportunity to react to the responders if he so wishes. We hope, however, to leave several minutes at the end of the end for questions from the audience and responses from any of our participants, and still meet our goal of ending this symposium between 4:45 and 5 o'clock p.m. Our two respondents today are members of our own faculty. The first respondent is Alice Fulton, professor of biochemistry and recently named director of the University of Iowa undergraduate honors program. Professor Fulton has an undergraduate degree and her PhD degree from Brown University. Our second respondent is David Depew, who is professor of communication studies and the project on the rhetoric of inquiry. He has also taught in the interdisciplinary program in literature, science, and the arts. Professor Depew attended undergraduate school at St. Mary's College of California and received his MA and PhD degrees from the University of California at San Diego. He has recently written a paper entitled, quote, Intelligent Design and Irreducible Complexity, a Rejoinder, which I believe is or will soon be published in the journal Rhetoric and Public Affairs. Gentlemen, we look forward to stimulating presentations and lively discussion. Would you please join me in welcoming Professor Michael Behe to the University of Iowa.
Thank you. As Ethan said, I have 30 minutes to speak not only, and the topic is criticisms and rejoinders, but I have to explain a little bit about what's being criticized and in the first place. So I'm going to speak a little bit about what my idea is. Sampling of the criticisms of the one or two people who have disagreed with me. Well, in 1859, of course, Darwin published The Origin of Species. And in it, he explained his idea how natural selection could drive evolution. And he also addressed a few what were thought to be problem cases. And one was the evolution of the eye. People knew that the eye had a lot of different parts, and if any of the parts were missing, then a person would be, his vision would be severely diminished, or he would be outright blind. So Darwin gave a little sketch of how he thought eye evolution could take place in a section of The Origin called Evolution of Organs of Extreme Perfection and Complication. And here, a little synopsis of what he said. It might be a little hard to see, but he says, well, some animals have very simple eyes, nothing more than a light-sensitive spot. Here are some photosensitive cells.
But, and this will detect light, but it can't detect which direction the light is coming from, because light coming from any direction will set off the cells. But if you put the light-sensitive spot into a little cup, now light coming from one direction, at least theoretically, can cast a shadow on the eye, and perhaps the organism can then sense which direction the light is coming from. And if you make the cup deeper and start to fill it with a gelatinous material, now you've got the beginnings of a crude lens. And so his explanation satisfies many people, but by standards of today's science, it's at the anatomical level. He didn't go into the molecular basis of all of these events he discussed. And that's not surprising, because molecules and atoms were not known, or they were theoretical entities at that time. People were not sure whether or not they actually exist. And Darwin wrote in The Origin of Species that how a nerve comes to be sensitive to light hardly concerns us more than how life itself originated. So he specifically turned down the task of describing where that light-sensitive spot came from. That was his starting point, because, again, he didn't have the tools to go into it. In the past century or so, and especially in the past several decades, science, of course, has made tremendous progress. And we have been able to look at those structures that Darwin had to take for granted. Let's ask ourselves, what has science shown about those light-sensitive spots? What is necessary for a cell to be sensitive to light? And here's what we now know about photosensitive cells. When a photon of light first strikes your retina, it isomerizes a small organic molecule called 11-cis-retinal, changing it into something called trans-retinal. And that's the first step in the chemistry of vision. And here's a little cartoon of the remaining steps. The retinal is attached to this little oval, which represents a protein called rhodopsin. When the retinal changes shape, it forces a change in the shape of the rhodopsin. That enables it to interact with another protein molecule called transducin, which had bound a small molecule called GDP, but that falls off and another molecule called GTP hops on. Now the rhodopsin, transducin, GTP complex acquires the ability to interact with another molecule in the cell called a phosphodiesterase, PDE, abbreviated here. The phosphodiesterase, when bound by these other molecules, acquires the ability to chemically cut a molecule called cyclic GMP, turning it into 5' GMP. Now, cyclic GMP binds to a number of molecules in the cell, one of which is this ion channel. The ion channel normally lets sodium ions into the cell and calcium ions, but when the cyclic GMP level is decreased due to its being cut by phosphodiesterase, the cyclic GMP bound to here falls off, the ion channel changes its shape and shuts down. That decreases the sodium concentration in the cell, which changes the charge across the cell membrane and eventually sends an electric current down the optic nerve to the brain. And when interpreted by the brain, that is vision. So that is Darwin's simple light-sensitive spot. And the point is that what Darwin and other scientists of the 19th century took to be simple starting points have turned out to be enormously complex. They did not anticipate, as far as I know, that a nerve or a cell would be such a complicated entity as we know it to be today. Well, okay, so it's complex. But how can we tell whether or not it can be explained by Darwin's theory? Well, it turns out that Darwin gave us a criterion by which to judge whether a system can be explained by his theory. He said if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Well, what kind of a system can't be formed by numerous successive slight modifications? Well, at least for starters, one that is irreducibly complex or has the property of irreducible complexity. Now, irreducible complexity is just a phrase that I made up to make people think that I'm smart. But it's actually a pretty simple concept. And it can be exemplified by reasonably well-known systems. Yes, this is a mousetrap. A mousetrap has a number of parts. It's got a platform, a spring, something called the hammer, which squashes the mouse. And when you push it over, you've got to stabilize the hammer in place until the mouse comes along, and there's something called a holding bar that does that. But the end of the holding bar itself has to be stabilized, and there's something called a catch. Now, the mousetrap is irreducibly complex because all of these components are needed for it to do its job. If you take away one of the components, you don't have a mousetrap that works half as well or a quarter as well as it used to. You've got a broken mousetrap. So things like this, irreducibly complex systems, are real challenges to Darwinian evolution because natural selection needs something to select. It needs a function that works in order to select it and hopefully improve it. But with an irreducibly complex system, like the mousetrap, there's nothing to select. If you start with a platform and hope to catch a few mice, and then add a spring and hope to catch a few more, you're going to be disappointed. The function of an irreducibly complex system doesn't appear until essentially the whole thing is put together. So something like this is a challenge to Darwinian type evolution. Well, mousetraps are very interesting, but the question is, are there any biochemical or cellular systems that are irreducibly complex also? And it turns out that yes, there are a number of them. And an example is shown here. Here's a little electron micrograph of some tissue, some lung tissue, and these little hair-like things are called cilia. And the cilia have the interesting property of being able to beat back and forth like this. And by doing so, they can either push a cell through liquid or push, if the cell is fixed, they can push liquid over the top of the cell. These big round things are called goblet cells, and they secrete mucus into the lining of your lungs. And the cilia beat in synchrony and push the mucus up towards your throat where you can cough it out along with the dust and other foreign bodies that collect in your lungs, and that helps to keep your lungs free and clear. But what does it take for a little cilium to beat back and forth like this? Well, investigations in the past several decades have shown that the cilium is a very complicated molecular machine. And here's a drawing of the cross-section of a cilium. And as you can see, it consists of a number of parts. It's got these double rings are called microtubules. It's got connectors between microtubules, several different kinds. One type is called a Nexon linker. It's got things called radial spokes, which lead in from the outer microtubules to the central inner microtubules. And it's got these other spokes coming off of the microtubules called dynein arms. And although that looks complicated, the cilium is actually much more complicated than that. It's got about 200 separate different protein parts to it. Well, that's what it looks like, but how does it work? Investigations in the past couple decades have shown that it works by something called a sliding fiber mechanism. And the microtubules are next to each other, and they're the fibers. And the dynein is something called a motor protein. What it does is hang on to one fiber, reaches over, grabs the other one, and pushes it down. And when it pushes it down, they start to slide relative to each other. And they would slide apart, except that the Nexon linker keeps them together. And as they start to slide, the Nexon linker although it was loose, and now it starts to become taut. And as it slides a little more and a little more, the strain increases until essentially it bends. And then it reverses and bends back and forth and back and forth. And that's how the sliding motion of the fibers is converted into a bending motion. Now, perhaps you can see that this is irreducibly complex as well. If it weren't for the sliding fibers, there would be nothing to slide. If it were not for the dynein motor protein, the whole thing would lie stiff and motionless. And if it were not for the necks and linkers, when they started to slide, the whole thing would fall apart, as it does in experiments when the necks and linkers are removed. So like the mousetrap, it's irreducibly complex, and like the mousetrap, it presents very big problems for a gradualistic Darwinian-style evolution. One more example I'd like to give you before moving on is in many ways like the first example in that it's an organelle for motion, but in other ways it's very, very different too. And this is called the bacterial flagellum. There's a little cartoon drawing of the bacterial flagellum taken from a popular biochemistry textbook by Voet and Voet. And the flagellum is quite literally an outboard motor that many bacteria use to swim. This part, made up of a protein called propellin, is the propeller, which contacts the water. And the bacterial flagellum has a rotary motor, one of only two known in nature, which rotates around and round. And the propeller is attached to the drive shaft by something called hook protein, which acts as a universal joint, allowing rotation around this end and this end as well. The motor to which the drive shaft is attached uses a flow of acid from inside the cell to outside the cell to power the turning of the drive shaft. The whole thing is kept in place and stopped from rotating so the motor doesn't rotate. There's something called a stator to lock it into the cell membrane. And there are various bushing proteins to allow the shaft to poke up through the bacteria membrane. And all in all, there are about 40 or 50 different proteins which are necessary either as parts of the flagellum itself or in the regulatory apparatus that turns the genes for these parts on and off. And again, you can see that if many of these parts are missing, the stator, the drive shaft, many of the proteins that have been identified as parts of this system, if they are missing, then you don't get a flagellum which turns half as fast as it used to, or a quarter as fast. Either it doesn't get made at all, or it doesn't turn at all. And so again, it's a big problem for Darwinian-style evolution. And in my book, I pointed out that there's a lot published in the biochemical literature on evolution. And for those not familiar with it, there's even a journal called the Journal of Molecular Evolution, which is set up to investigate how evolution occurs at the molecular level. Now, you can't read the titles of these papers, but suffice it to say that in the biochemical literature on this, essentially all of the effort is focused on what is called sequence analysis. Now, proteins and nucleic acids are both made up of building blocks of amino acids for proteins and things called nucleotides for nucleic acids. And an interesting thing to do is, for example, take a protein from one species and an analogous protein from another species, say hemoglobin from horse and hemoglobin from dog, and compare the sequence of amino acids in the protein. And ask yourself, is the amino acid in the first position of this protein the same or different than the amino acid in the first position of this protein? How about the second position, the third position, the 50th position, and so on? And that's a very interesting thing to do, and comparing those sequences can give you a good idea of how related the species are. But comparing protein sequences does not give you any insight, or does not tell you how complex molecular machines like the cilium and flagellum and other things could have been put together step by step. Let me give you a little analogy. It's kind of like comparing the bones in the forelimbs of a dog and a horse, and noticing that there are similar number of bones and they're at the same relative positions. And that's interesting, and it gives you a good idea of how related they are. But comparing the positions of those bones does not tell you where bones came from in the first place. In order to do that, you've got to do experiments, you've got to build models and other things. It turns out that in the entire history of the Journal of Molecular Evolution, there have been no papers published which actually build detailed models to see how complex molecular machines like the psyllium flagellum and other things could have been put together step by step. There's been a lot of good work done and a lot of sequence comparisons, but they don't address the question that we're interested in here today. And that, in my opinion, that attitude is pretty widespread in the literature. If you look at other journals besides the Journal of Molecular Evolution, the story is pretty much the same. And in the textbooks as well. In the textbooks, students are taught Darwinian evolution, and a good example of that is this little cartoon taken from Voet and Voet, which kind of summarizes the orthodox idea of how life came to be and subsequently diversified. And as you can see, the top third here shows a little volcano and lightning and some gases floating about, and that's how life started. And shows a kind of schematic of DNA coming out of the origin of life ocean and going into a bacterial cell, and that's how life developed. And here's the bacterial flagellum here. And at the bottom here we have the Garden of Eden, showing a bunch of different species produced by evolution milling about, and that's how life diversified. But if you look in that textbook for real solid answers on how any of these processes could have occurred, you don't find them. They're missing. And so the problem is that it seems that imagination has taken over much of the field of explaining how complex molecular machines. If you look at the drawing of the bacterial flagellum here, from this perspective, it doesn't look like much of a problem at all. But when you go up close and examine the parts of it, you see that there are reasons to think that Darwinian processes might be inadequate to producing something like that. Now, what I have said to this point is not really new. A number of critics of Darwinism have appeared, people like Stuart Kaufman at the University of Pennsylvania, Lynn Margulis at Massachusetts, and they've said that they agree that these things look like they are beyond Darwinian explanation and that there's not much work been done on them. Where I differ from those other folks is in the explanation I have advanced for how these things came about. And I said, if you look at them, they certainly look to be designed. They look like they were purposely designed by an intelligent being. And that has gotten a bit of attention. And many of my critics have said, well, this Behe fellow is an admitted Christian, and he's been seen going into churches on occasion. And therefore, this idea of design is a religious conclusion, not a scientific one. And I'm letting my religious views interfere with my science. Well, not surprisingly, I disagree with them. I think the conclusion of design is a completely empirical conclusion, based solely on the physical evidence itself, along with an appreciation for how we come to a conclusion of design. And to see how we do, in fact, conclude design, let's look at the following. It's a Far Side cartoon. We have a bunch of jungle explorers, and the lead explorer has been strung up and skewered. And the fellow with the glasses turns to this guy and says, "That's why I never walk in front." Words to live by. Now, everybody in this auditorium looks at this cartoon, and you immediately recognize that this was designed. It wasn't an accident. His death was intended. How do you know that? Is it a religious conclusion? No, of course it's not a religious conclusion. You realize that this was designed because you see a number of different parts interacting with each other to perform a function that none of the parts by themselves perform. Essentially, you see irreducible complexity, much like the mousetrap. So, this is really not hard to see. When you can illustrate your main point with a Far Side cartoon, we're not talking quantum physics here. But even though it can be seen easily, it can also be treated as a formal academic subject and investigated. And that's what William Dembski has done in a book that has just come out from Cambridge University Press. He's a mathematician and philosopher, and he asked the question, what is design? How do we conclude design? How do we differentiate it from chance and natural law and so on and so forth? And for those of you interested in that question, I highly recommend his book. Okay, and now let me, in the last 3 minutes, well, now the last 10 minutes or so, what have other scientists had to say about this? The book has been controversial from the start, which is good. If you write a book, you want it to be controversial because that means people will notice it and think about it. Well, it turns out that the scientists, virtually all scientists who have reviewed the book, have agreed with me that these systems that I write about are enormously complex and unexplained, currently unexplained. For example, James Shreve, who's a science writer and paleontologist, writes in the New York Times, Mr. Behe may be right that given our current state of knowledge, good old Darwinian evolution cannot explain the origin of blood clotting or cellular transport. James Shapiro, a professor of biochemistry at the University of Chicago, writes, there are no detailed Darwinian accounts for the evolution of any fundamental biochemical or cellular system, only a variety of wishful speculations. Jerry Coyne, a professor of evolutionary biology at the University of Chicago, writes in Nature, there is no doubt that the pathways described by Behe are dauntingly complex and their evolution will be hard to unravel. We may forever be unable to envisage the first proto-pathways. Well, that's my favorite one there. In case, do you have trouble seeing it? And some people go even further.